Hola, Team Bio. Okay, today I am going to be talking about aerobic cellular respiration on this screencast, so if you have your roadmap here. Yesterday we talked about anaerobic cellular respiration, which included glycolysis and two types of fermentation. And today, again, it's going to be aerobic cellular respiration. I'm going to try and keep it short. I am not going to cover glycolysis again because I just did that yesterday. So if you have questions about glycolysis, go ahead and watch the previous really long screencast, or come ask me in class. Today we're just going to be talking about the prep steps and the Krebs cycle. And this electron transport phosphorylation, I'm not going to teach that to you, and we are going to figure it out together in class tomorrow. Okay, so let's just jump right into it. So starting out with glycolysis, we're going to skip that, but we're going to talk about what comes out. And as you remember from yesterday, at the end of glycolysis, we end up with 2-pyruvate molecules. Okay, so 2-pyruvate are the end product of glycolysis, and you should know, so everything that I talk about now, I'm just going to talk about what happens to 1-pyruvate molecule, but you should know that for a complete digestion or breakdown of glucose, this all happens twice, because remember, one molecule of glucose produced 2-pyruvate. But I'm just going to be talking about what happens to one. So the first thing that happens to the pyruvate after it leaves glycolysis is it enters the preparatory steps, or the prep steps. And in the prep steps, which are also taking place in the cytoplasm, this molecule, this pyruvate molecule, is going to be oxidized once again. So here I'm just going to grab 1-pyruvate, copy it below. So 1-pyruvate molecule plus something called coenzyme A. Oops. And a coenzyme is something, it does exactly what it sounds. It works alongside an enzyme to let a reaction take place. So coenzyme A is going to let this molecule undergo a reaction, this pyruvate molecule. So 1-pyruvate, 1 coenzyme A, and 1 NAD plus. Franklin, quiet. Franks, relax. Sorry. Is all going to form this compound called acetyl-CoA. So this is acetyl-CoA, ACTYL-CoA, which is like a combination of, we stuck coenzyme A onto pyruvate, but you'll notice it's only 2 carbons long. So what happened to the other carbon? Well, it broke off and it's going to be excreted as CO2. So this is our first molecule of CO2 that we've seen produced in humans. So when we breathe out, it's actually some of the CO2 that you're breathing out is coming from the prep steps. And then additionally, we also get 1 NADH. So NAD plus over here grabs an electron and some free hydrogen. It grabbed the electron from the pyruvate molecule during this reduction oxidation reaction, and it was formed into NADH. And again, this is an important molecule. I'm not going to talk about it today in the screencast, but you'll see why it's so important when it comes to the step of oxidative phosphorylation. Okay, so here are the prep steps, and from the prep steps, we're actually only going to take one piece, and that is this acetyl-CoA. This molecule that we created right here is the only thing that's going to move forward into the Krebs cycle, which is going to take place in the mitochondria. So this will move to the Krebs cycle, which is also called the citric acid cycle. You'll see it written as either form kind of like the Calvin cycle, which has many names. The Krebs cycle has a few names. And these are the two most common. And this all takes place in, remember, the mitochondria. So the Krebs cycle is happening in the mitochondrial matrix, which is the very interior of the mitochondria. Okay, and I just want to emphasize again that we had 2 pyruvate that came out of glycolysis. So remember, I only wrote 1 pyruvate, 1 coenzyme A, 1 NAD+, 1 acetyl-CoA, 1 CO2, 1 NADH. But remember, this is all doubled. This happens twice because there are 2 pyruvates that enter each prep step. Okay, so moving on to the Krebs cycle happening in the mitochondria. Okay, so we start out with acetyl-CoA and also this 4-carbon molecule that you all know and love. It's oxaloacetate. And it's important to keep in mind that oxaloacetate it's a low energy molecule. So I'm going to indicate that. I wish we had orange on this. I don't. Okay, so this is low energy. And it joins up with acetyl-CoA, which is going to, I don't like that arrow. So acetyl-CoA joins with oxaloacetate, and acetyl-CoA is a relatively high energy molecule, and they're going to make a 6-carbon molecule called citrate, or citric acid. So here's our citrate. Citric acid. I think you can see now why this is called the citric acid cycle. Okay, so, and I'm going to just grab this, move it over a little bit. This citrate, the citric acid, is a very high energy molecule, and it can be oxidized, meaning it can lose electrons, and it can donate high energy electrons to, you know what, electron carriers. So the first thing that happens after citrate is made in our citric acid cycle is that NAD plus flows in, and out comes, actually I'm going to do this in another color. So out comes NADH. Remember, our high energy or high energy electron carrier. And in this process, as this citrate is being oxidized and NAD plus is being reduced, we also break citric acid apart and a molecule of CO2 is released. And this will be exhaled, just like this CO2 here also will be exhaled. Okay, so now we have a 5-carbon molecule, which I'm not going to give you the name of, but the same thing happens again. So NAD plus comes in. I'm going to copy this, paste it down below, and NADH is produced. And in this process, a molecule of CO2 is produced and will ultimately be exhaled. Okay, and then that leaves us with a 4-carbon molecule. So again, just copy this 5-carbon molecule, but only grab 4 carbons, and a 4-carbon molecule is left. And this 4-carbon molecule is also it's still very high energy, and it can still do a lot of work before it's converted back into oxaloacetate, which is our low energy molecule. Remember, it's a low energy molecule. So what happens? Well, the first thing that happens is an ADP and a free phosphate come in to the cycle, and an ATP is produced. And next, another electron carrier that you have not seen before. It's called FAD. FAD comes in, and out pops another high energy molecule, or a molecule carrying a high energy electron, and that's FADH2, and actually it carries 2 high energy electrons, so I should say that. And then the last thing that happens is NAD plus comes in, and once again NADH is created. So throughout the course of this cycle, we slowly oxidize our citrate, our high energy molecule, until ultimately at the end we're left with a low energy oxaloacetate. And then the whole cycle repeats itself. So remember, 2-pyruvate were created in glycolysis. And then 2-acetyl-CoA come out of the prep steps. So for every molecule of glucose, there's actually 2 turns of the Krebs cycle. And so for every molecule of glucose, 2 NADH will be created here, 2 will be created here, 2 ATP, 2 FADH, and 2 NADH. So if we were to do our total accounting, which we should do just really quickly. When we start out with 1 molecule of glucose, 1 glucose, in glycolysis, we get 2 NADH and 2 ATP. Remember, this is on net, because we spend 2 and then we make 4, so on net we get 2. In the prep steps, we get 2 NADH per molecule of glucose. And in the Krebs cycle, per molecule of glucose, you get 6 NADH, 2 FADH2, and 2 ATP. And then, again, we also get 2 CO2 here, but that's kind of just a waste product, and here you get 4 CO2. So, a big question is, the whole purpose of cellular respiration is to make ATP, and yet, so far, we've only created 4 ATP total. So the next big question is, how does the rest of the 34 to 36 ATP, no wait, that was the 32 to 36, no, 32 to 34, no. How many total? 32 to 34 are created in the electron transport thing. So how do those next, the vast amount of the ATP get created? And that's something that I'm not going to tell you, but we are going to explore tomorrow. Okay, 14 minutes, not terrible.